My name is Hong Liang, and welcome to the Forensic Entomology Subspecialty. So I bet some of y'all don't really know what in the world is entomology, right? Okay, don't worry. We are here to explain in this video. So forensic entomology is the study of insects and arthropods, such as flies or spiders, in a criminal investigation. Right from the early stages, insects are attracted to the decomposing body and may lay eggs in it. Sounds kind of girly, right? I know. So I think we can all relate to this while we see many flies hovering around those fresh meat at pasar, right? So these insects will lay eggs at decomposing bodies. And by studying these larvae, forensic entomologists can estimate the cause of death and also the time of death, or another fancy way of calling it,
minimum post-mortem interval MPMI
of the body. So there are 5 stages during decomposition of body, which are fresh, bloated, active decay, post-decay, and lastly, dry stage. Among these stages, fresh and bloated stages play a big role in estimating the time of death, as insects are most attracted at these early stages. Okay, now enough of me talking. I'll pass to our Miss Shannon to explain more on these examples of forensic insects.
Thank you, Liang. That was a really good introduction on forensic entomology. So, what are the examples of forensic insect, you wonder? In general, flies. Example, blowflies, flesh flies are the first to visit a corpse to lay their eggs. Once eggs hatch, the fly larvae begin feeding on tissues. Besides that, beetles. Example, carrion beetles, hem beetles, rove beetles are also commonly associated with decomposition and can be used in the estimation of the PMI. Some insects can also interfere with the estimation of this PMI. For instance, ants, or also its fancy name, Formicidae. So these ants present at the corpse may eat fly eggs and larvae, hence slowing the decomposition rate of the corpse. Now, Liang will tell you what are the key differences between species of flies and how to differentiate them.
All right, thank you, Shannon. So insects that are mostly found in forensic investigations are true flies or Diptera. So under the category of true flies, we have 3 main families, which are Calliphoridae, blowflies, Sacrophagidae, flesh flies, and lastly Musidae,
houseflies and their relatives.
I'm sure you may have seen some of these species of flies before. Now let me show you some pictures of each flies. So this is Calliphoridae, the blowflies. It is shiny and with a metallic blue, green, or black color on their body. Looks familiar, right? Now next, we have Sacrophagidae, the flesh flies. As we can see, it is quite different from the blowflies before. It has black and gray stripes instead of the shiny one. And lastly, we have our Musidae,
houseflies and their relatives.
It is quite same with flesh flies, except if
they are dull and smaller than Sarcophagidae.
Interesting, right? So blowflies and flesh flies may arrive within minutes following the death of a body, whereas houseflies only arrive at bloated stages of decomposition. Okay, next, I'll pass the video back to Shannon again.
Once again, thank you, Liang. So, what are the importance of forensic entomology, you ask? Forensic entomology is important in determining the elapsed time since death. Insect evidence may also show that the body has been moved to a second site after the death. For instance, some insects on the body may be native to the first habitat and not the second. This will show that not only was the body moved, but it will also give an indication of the type of area where the murder actually took place. And it is also important to tell that if the body has been disturbed by any animals or by the killer returning to the crime of the scene. When this happens, it may disturb the insect cycle And the entomologists may be able to determine not only the date of death, but also the date of the return of the killer. However, the primary importance of forensic entomology today is to determine elapsed time since death. Now, Bhavanish will briefly explain to you on how insect samples are collected and preserved from crime scenes.
Thanks, Shannon. So by now, I'm sure all of you have a rough idea of what forensic entomology is and how important it can be in solving crimes. So as mentioned before, specimens of forensic entomology play an important role in solving cases, especially when there's a carcass involved. So obviously, you would want to collect the specimens from the carcass so that further investigations can be done. So essentially, there are three main details that forensic entomologists need to pay attention to when they are collecting samples and preserving them at the crime scene. So these include collecting the live and preserved specimens, recording the conditions of the scene, like its surrounding temperature and weather conditions, and finally, transporting the specimens to the lab while maintaining the chain of custody. So first, let's talk about specimen collection. So obviously, the first thing you need to do before entering the crime scene is to make sure you wear the appropriate attire. So this is done to avoid contamination of the scene. Next, you should introduce yourself to the investigating officer or the person in charge of the crime scene. Then you need to establish the area for collection of specimens. So always be sure to bring along your forensic entomology kit. This kit should have all the necessary equipment such as forceps, jars, vials, hand net, and other important tools for you to carry out specimen collection or preservation. You will also need to collect 3 or 4 soil samples from under the ground where the carcass is. To preserve the soil samples, place it in the refrigerator, but make sure not to freeze them. The same goes for the collected specimens. They should be placed in a cooler or in the fridge. Remember to never freeze them. So it is advisable to collect about 2 dozen big maggots and fly pupae, but make sure to separate them. Fly pupae look like long red to dark brown seeds, so be sure not to miss them when you're collecting samples. I will attach an image here so you can have a quick look at how it looks like. And when you're collecting maggots, make sure you separate them based on their characteristics. So for example, if you're collecting hairy and smooth maggots, make sure they are placed in 2 separate jars. And if you're preserving the maggots, place them in 70 to 90% ethyl alcohol. So once you're done with specimen collection, you need to record the condition or ecological features of the scene. So things that you should be recording include condition of the carcass, temperatures of the body, larval masses, and temperature of the surrounding. You also need to get information regarding the weather prior to the discovery of the body and during the present time. Finally, after recording the data and collecting the samples, you need to label all your jars and other packages that contain your samples or data. You need to write down your name, the person receiving it, the date and time of sample collection, and other related information. Then at last, you must seal each sample securely to guarantee the chain of custody. I know that was a bit technical, but I hope you guys understand and familiarize with the important steps that need to be done when collecting and preserving samples at the crime scene. So next comes the fun part. We want you to rear your own live samples of larvae at home using the items provided in the forensic toolkit. So for this, my teammate and my friend Iza will now show you guys how to rear larvae all by yourself. So I hope you have fun.
Okay, thanks to my friends Shannon, Liang and Baba. So hi, Assalamualaikum, I'm Iza. Now I'm going to share with you guys how to conduct a larva rearing inside your house. Also before that, don't forget to bring along the home lab kits that we already provided to you. And yeah, so let's continue watching.
Okay, for the preparations of the container, we need a piece of paper, a pair of scissors, a pin, piece of tissue paper, and a forceps. First, cut the paper into small pieces. Then, crumple it into a small ball. We're going to make it as a base inside the container. Then, place a piece of tissue paper on top of the crumpled paper. Okay, next. Punch holes in the lid to allow the air flow by using a pin or a needle. After that, cover the mouth of the container with a piece of tissue paper. This tissue paper under the lid is important to provide a dark and damp conditions for the lava. Okay, next, to prepare the baits. As mentioned before by my friend Liang, flies are attracted to the decomposing body. So basically, an adult fly feed and harvest their larvae on decaying material because they can detect the odor produced from the rotten material. So to attract the flies, you can use either raw food or leftover food. This part will be a bit longer for you to wait, so make sure to put the bait at the most possible place to find the fly. Most often they can be found outdoors. Okay, then transfer the eggs into the prepared container and make sure to put along with it enough food for the larva once they already hatch. Now all you have to do is to wait because they will begin to hatch within a day. After 24 hours or after a day, the larvae will hatch out and we usually see them in the first stage while they are still 3 to 9 millimeter long and whitish in color. So basically, they will eat their way through the decaying food before pupating and then becoming an adult fly a few days later. So if you guys want to see further how the larvae becoming flies, you can continue the rearing up until 1 week.
Let's look at the life cycle of a fly to get a better understanding. The life cycle of a fly starts with an egg stage, moving on to 3 different larva stages, and going through a pupa stage before emerging as an adult. The first larval stage having the length of 3 to 9 mm long after 24 hours. The second larval stage having the length of 10 mm long after 2 to 3 days. And the third larva stage having the length of 14 to 16 mm long with greatly increases in size after 4 to 5 days. In the third stage, they stop feeding and the larva starts to migrate away from the moisture because they like dry and secure areas away from the wet food source to pupate. Lastly, the adult flies emerge from pupa cases after 6 to 8 days. I hope you guys enjoyed watching this video.
Thank you.